Thank you, Maria. Welcome, everybody, to today's meeting of the ICOC and Application Review Subcommittee. Maria, will you please call the roll?
Dan Bernal, Leandra Clark-Harvey, Anne-Marie Dulege.
Yes.
Isabel Dudon.
Here.
Mark Fisher-Colbury.
Here.
Fred Fisher.
Good morning.
Elena Flowers. David Higgins.
Here.
Steve Julesgard.
Present.
Rich Lahara.
Here.
Christine Miskowski. Lauren Miller-Rogan.
Here.
Adriana Padilla.
Here.
Joe Panetta. Al Rolette.
Present.
Marvin Southard.
Here.
Jonathan Thomas.
Here.
Art Torres.
Present.
Carol Watson. We have quorum. And
This
I want
is
to note,
Leandra. I
Yes.
think you missed my present. You moved really fast. Sorry. I'm here.
oh no, thank you. And I noted you as present. Thank you so much. Larry Gold, I want to note that other board members who have joined, Larry Goldstein. So thank you, and JT.
Thank you, Maria. We will now move into the meeting of the Application Review Subcommittee. Have one action item with multiple parts today: consideration of applications submitted in response to discovery stage research projects program announcements, or the so-called DISC 2. Begin with a presentation from Dr. Sembrano. Gil?
Okay, thank you, Mr. Chairman. So hopefully you can see the presentation. Let me just put it in full screen. Okay, good morning, everyone. I'm going to present to you the recommendations from the Grants Working Group related to our latest cycle of the DISC-2 program. And before we start, as always, we want to remind everyone, including ourselves, about our mission and our goal and why we do all of this, which is to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. And so the DISC-2 program is part of our recurring opportunities that we offer throughout the year. So this particular one happens twice a year, and it is at the early stages of bringing new ideas to develop a single product candidate. The specific objective of the program is to promote the discovery of promising new stem cell-based and gene therapy technologies that could be translated to enable broad use and ultimately improve patient care. And so we are looking here for projects that will uniquely enable human stem cell progenitor cells in some way, or are uniquely enabling for the advancement of stem cell-based therapies, or for developing a gene therapy approach. All right, and the types of products that can come into this type of competition include therapeutics, that's what we see most of, but also diagnostic devices and tools. In both cases, what we're looking for generally align with looking for developing a single candidate or a single prototype that is identified by the end of the award, the development of a target product profile, which is sort of an idealized summary of what they hope to achieve with their product if it's successful, and then a proof of concept of some type. So for a therapeutic, showing that disease modifying activity occurs with the therapeutic, that means it has an effect on the intended disease, or for a tool, for example, some kind of proof of concept that the tool works as intended. And so this DISC-2 candidate discovery program fits in within our pipeline, and just here simply to show you the approximate time. So 24 months is what we currently allow for the DISC-2 program. That is going to change to 3 years in the next round. For the translational programs, those that achieve a single candidate can qualify for TRAN. That's another 24 to 30 months to get them to a pre-IND meeting. And then subsequent to that, if they are successful there, they can qualify for potentially a CLIN 1 award to do IND enabling work, and that's another 24 months. So here simply to say that at this stage, applicants are going to still be at least 6 years away from getting to the clinic for these programs. So still pretty early phase. All right, so as it pertains to the review itself, we conduct the review of these applications in a two-stage process, which we often refer to as positive selection. So if you hear that term, it's related to the first stage of the review. And so this happens when we have a total number of applications that come in that exceeds the capacity of the grants working group to review in a single session. So that's pretty common for most of the discovery competitions that we have. So in this first stage, the grants working group members, including the patient advocate and nurse board members, as a panel conduct a pre-review of applications and select which ones to advance to a full review. Of those that are not selected, the Serm president and Serm team look and examine to see if there are any additional ones that would merit a full review, and the remainder are not considered. So for this particular round, we had a total of 75 eligible applications that were submitted. We had 13 that bypassed this process because anything that scores between an 80 and an 84 doesn't have to go through this process. And we ended up with a total of 54 that advanced to the full review. So the scoring system for these applications is based on a scale of 1 to 100. Anything that receives a score that's greater than 85 is recommended for funding. Anything that receives a score of, and this is wrong, sorry, 80 to 84 is not recommended, but those get to then bypass the positive selection process if they come in the next round. And so reviewers, when they score, know this, and so they generally try to score applications between 80 and 84 if they want to see it come back and feel that the revisions are likely to be minor. Anything that scores between 1 and 79 is not recommended for funding. And so this is all based on the median score from all individual GWG scores. The review criteria that are utilized to give those scores are based on these 5 questions. Does the project hold the necessary significance and potential for impact in terms of the value that it offers, and is it worth doing? Is the rationale sound? Is it well planned and designed? Is it feasible, including whether they have the appropriate resources and a qualified team? And does the project address the needs of underserved communities? So we get to this slide, which is just a reminder to all our board members who have a conflict of interest. So if your name is on this list, just remember that you may have a conflict with one application, which means you should refrain from participating in voting or discussion until the very end. All right, so here are the recommendations from the grants working group as it relates to this particular cycle. So we had, as mentioned, 54 applications that were reviewed by the panel. 17 of those applications were recommended for funding because they received a score of 85 or greater. And so that total applicant request from the 17 applications is about 22 million. The funds available in the discovery pillar is almost 58 million, but that includes the DISC 0 amount as well. So there's intentionally more money here because we still have another competition for the DISC 0. That's the new basic early foundational biology that is yet to come to you. Let's see. And then 37 applications were not recommended for funding under this cycle. Some of the applications that are reviewed and scored qualify for what is called a minority report. So under Prop 14, any application that is not recommended for funding by the grants working group, meaning that it scored below 85, but which had 35% or more of the members score to fund the application, must include a minority report. And so the minority report is included in the review summary and provides a brief synopsis of the opinion of the reviewers that scored that application 85 or greater. And so for this round, we have 4 applications that qualified for a minority report, and I will review those individually and go into more detail. Before I do, I want to just mention the Serm team recommendations as it relates to this cycle. And I want to point out that when we have applications, particularly those that qualify for a minority report, the Serm team examines the applications just to determine if we have any recommendation one way or the other for these. Generally, what we do is we recommend that applicants revise their application to address reviewer concerns and submit in the next cycle. We offer the DIS2 twice a year with the goal of bringing, and as noted earlier, there are many resubmissions that come and into the cycle, many who scored between 80 and 84 that get to bypass that initial stage of review and are able to come in. So the next cycle deadline is going to be August 2nd. So anyone that does not get funded in this cycle can certainly come in August 2nd. And those that scored between 80 and 84 get to bypass that first stage. And there's 11 applications that qualify for that. Now, in some cases, when we review the minority reports and those applications that are near that funding line, there are applications that otherwise are meritorious, but have concerns that really can't be addressed in a resubmission, and where those concerns would not necessarily impede achieving the goals of the DIS2 program. And so when we come across those types of applications, we may make a recommendation to go ahead and fund those. And so we have 2 that met those criteria for us. So those are application 13510 and 13475. And so I will go into more detail about each of these in the next few slides. Okay. All right. So the first one is related to application DIS2 13510. This application received a score of 84, and we had basically a tie. There were 7 members that scored 85 or above, and 7 members that scored below 85, with a range of 80 to 90. So the title of this application is a hematopoietic stem cell-based approach to treat HIV, employing CAR T cells and anti-HIV broadly neutralizing antibodies. So clearly this is for an indication of HIV infection. The product is a cell and gene therapy that basically involves hematopoietic stem cells that would be transplanted in order to produce CAR T cells that would act against HIV, as well as produce B cells or plasma cells that secrete broadly neutralizing antibodies to also act on any free HIV virus and hopefully surveil and maintain the latent reservoir at bay. So that is that application. So let me present to you the summary of the minority report. So the minority report for this application states that 7 grants working group panelists scored the application 85 to 90, 7 scored the application 80 to 83. Overall, most of the panelists voted yes on whether the application had met each of the 5 review criteria, and the success of the project would address an unmet need. The rationale is sound, the project is well planned, and it's feasible and addresses needs of the underserved. Those who scored between 85 and 90 were optimistic about the dual transgene approach using both the CAR T and neutralizing antibodies, noting that CAR T therapies developed to date for HIV had shown some effectiveness. These high-scoring panelists had similar thoughts about the risks of failure as lower-scoring panelists, but this combined with enthusiasm for the potential payoff for patients. One high-scoring panelist wrote, "The project is a preclinical proof of concept in relevant in vitro and in vivo models. It represents an important stage bridging discovery and translation." The panelists who scored 80 to 83 to not recommend for funding noted the concerns that the therapeutic HSCs would not properly differentiate into all types of mature T cells in vivo, therefore limiting the safety and efficacy of the therapy. So this application was a resubmission, meaning that it's been seen before by the panel. And the concerns from the reviewers relate to the potential safety of a future candidate if the vector and construct would ultimately interfere with appropriate maturation of T cells. So that was the driving concern. Now, a resubmission may not be able to address this specific concern, as such work might be more appropriate for a later stage of research when the final candidate has finally been achieved, so maybe at the translational stage. So for this particular stage of research, Serm's team believes that the applicants have an appropriate proposal that wouldn't necessarily be improved by trying to address that concern and would not impede achieving the goals of the DIS2 program. So for this one, we are recommending funding. The next application is DIS2 13475. This received a score of 84. The number of 85 or above voters was 7 versus 8 who scored below. The title is Developing a Gene Therapy for Dominant Optic Atrophy Using Human Pluripotent Stem Cell Derived Retinal Organoid Disease Model. The disease indication is an inherited genetic disease called dominant optic atrophy. It is a gene therapy approach that would overcome the gene defect. The proposal itself is looking to develop the appropriate gene therapy and test this in human retinal organoids in order to determine if they have an effective therapy. All right, so the minority report for this one reads as follows. 7 panelists gave the application a score of 85, recommended for funding. 8 panelists scored from 79 to 84. Nearly all grants working group panelists agreed that the application met 4 of the 5 review criteria. The success of the project would address an unmet need. The project is well planned. The project plan is feasible, and the proposal addresses needs of underserved groups. However, the grants working group was divided, 9 versus 5, on whether the applicant met the criterion to the provision of a sound rationale for the therapeutic approach. The 8 grants working group panelists who scored the application 79 to 84 were uniformly concerned about using a gene replacement or augmentation therapy for a dominant Mendelian disease. In a dominant Mendelian disease, such as dominant optic atrophy, or DOA, a person carrying a single mutant copy of the 2 copies that a person has in their genome will have the disease. The mechanism of dominance varies from disease to disease. It can either be haploinsufficiency, meaning there's only that one healthy copy is insufficient for health, or a dominant negative where the mutated copy is actively harmful. And so the mechanism of Mendelian dominance in this particular indication is not known. The 7 panelists who gave the application a score of 85 agreed that the rationale for gene replacement will be weaker if the disease indication has a dominant negative mechanism. However, these panelists were optimistic based on the admirable amount of preliminary data from the patient-derived retinal organoids transduced with the candidate therapy in which the cell type that degenerates in DOA appeared to show recovery. So this also is a resubmission, meaning the grants working group has looked at this application now twice. The concerns from reviewers relate to, as explained, the possibility that DOA might be a dominant negative mechanism and therefore making the therapeutic much less likely to work. Now, a resubmission would not be able to really address this concern, but studies performed under the proposal would certainly determine if the therapeutic approach would be effective or not, even in the absence of knowing the underlying dominance mechanism. So therefore, Serm team believes that the best way to know is to advance this project, and we would recommend this one to move forward as well. All right, the third minority report is for application DIS 2-13413. This also received a score of 84. There were 6 members who scored 85 or greater, 8 that scored below 85, with a range of 70 to 85. This application is entitled In Utero Treatment of Duchenne Muscular Dystrophy with Non-Viral Gene Editing for patients with Duchenne Muscular Dystrophy. So this is generally a gene therapy approach. More specifically, it's a lipid nanoparticle mRNA complex that would be introduced in utero and hopefully reach all different affected organs and tissues in order to correct the dystrophin mutation in such patients. And so the minority report for this one is as follows. So this 6 panelists gave this application a score of 85, 8 panelists scored between 70 and 84. The great majority of panelists voted yes as to whether each of the 5 review criteria were met. Overall, reviewers in favor of funding the application thought there was a significant unmet need for Duchenne muscular dystrophy treatments and thought that the approach of using lipid nanoparticles for targeted gene editing in utero made sense. These reviewers also thought the preliminary data was strong, the proposed project was highly innovative, and the team was well qualified to complete the work. One supportive reviewer noted that the dependency on AIM-3 on the success of AIM-2 was a potential risk for the project, as no alternatives were presented. Greatest divergence between the high scoring and low scoring panelists is in the comments for criterion 4, feasibility of the project. Reviewers in favor of funding thought the timeline was feasible and the team was qualified. Reviewers not in favor of funding said the project was too ambitious, uncertain, or had too many milestones. No reviewer expressed doubts of the proposed team. So now for this application, this is a first-time applicant, so it's not a resubmission. The applicant, based on the criticisms, has the opportunity to address the concerns in a revised application by reevaluating the proposed activities and design of the project in order to make it conform better to the goals of the DIS-2 program. Now, the applicant would, because of the score they received, would bypass the first stage of review. And then the next cycle of DIS-2, as I mentioned earlier, does offer an additional year of funding, as well as more funds for that additional year. That would help, particularly in this case, address the concerns related to having too many activities. So basically, it would give the applicant more wiggle room to work with. So we're not recommending this particular application for funding. The last minority report relates to the DIS-2-13442. This one scored 83 with 6 members scoring 85 or above and 8 scoring below. The title is Microgel Encapsulated IPS Derived Notochordal Cells to Treat Invertible Disc Degeneration and Low Back Pain. So this is for a disease indication where invertible disc degenerates with the hope to use a cell therapy that would allow them, such patients, to gain some recovery of tissue and hopefully avail them of low back pain, as well as of any further damage to their disc. All right, so the minority report for this one reads as follows. 6 panelists gave this application a score of 85 to 86. 8 panelists scored 80 to 83. All 14 scoring grants working group panelists agreed with a yes vote that the application meets 4 of the 5 review criteria. Success of the project would address an unmet need. The rationale for the approach is sound, the project plan is feasible, and the proposal addresses needs of underserved groups. Panelist votes were split 9 to 5. Am I reading the right one? Yeah. On whether the application meets review criterion 3 for a well-planned and well-designed project. Panelists who scored the application 80 to 83 emphasize the importance of additional preliminary data demonstrating the cell product functionality in its final formulation. The panelists that scored 85 or 86 were favorably impressed with the study rationale and the applicant's published study results showing derivation of notochordal cells from iPSCs. These panelists also made positive comments about the strength of the preliminary data generally and the feasibility of the project. High-scoring panelists acknowledged the need for supportive data from studies using the final candidate product, but thought that the product plan put the applicant in good stead to collect this data during the project period. So here we have an application. This is not a resubmission. This is a first-time application. The applicant has, based on the critique, concerns that could be addressed in a revised application by providing additional data, as well as responding to some of the concerns raised by reviews, including the formulation of the candidate. So we are not recommending this application for funding. And that concludes the minority report summaries. If you give me just 1 second, I will put up the Excel sheet that shows the all of the applications. So this is also shown in your materials, but this is the rank order of all the applications. There are 17 in green that are recommended by the grants working group. The 4 applications that received a minority report, the 2 that the Serm team is recommending are these 2 here, 13510, 13475, and then below all that are the ones that scored, that did not receive a minority report or scored below 80. All right, so Mr. Chairman, back to you.
Thank you very much, Gil. So here's how this is going to work, everybody. The first thing I'm going to do is ask if there are any motions to move any of the projects currently in the not recommended for funding list up to the recommended for funding group. If there are none, we will proceed immediately to entertain an omnibus motion to approve those in the top tier. If there are some that are moved to move up to the top tier, we will first vote on whether or not to move them up and then proceed to the omnibus motion on all of the grants in the top tier. And following that vote, we will close out the voting by a vote to not recommend for funding the remaining projects in the not recommended for funding range. So the first,
JT,
yes.
really quickly, just as a reminder, you might want to also ask if there are any in the funded that people feel should not be funded. So that's an extra step. Sorry about that.
Thank you. Yes. Okay, so first question. Would anybody like to move to move any of those projects currently not recommended for funding up to the recommended for funding tier?
I move that we follow the staff recommendation and move the two that they recommend to the funded category.
Okay, moved by Marv Southard. Do we have a second?
I second. Second.
Al, that was you? Got a little,
Yes, it was.
you got a little feedback there. Okay, thank you. Okay, is there any discussion by members of the board on this motion?
Anne Marie has her hand
Anne-Marie.
raised.
Anne-Marie and then Fred. Anne-Marie.
Great. Thank you, JD. First, Gil and the Serm team, congratulations again for an excellent process. I won't belabor the points, just go right to the point and maximize our time and expertise and your time and expertise. For these two that are recommended, I'm a little puzzled by the fact that the discussion from the DWG was about this, if I understood correctly, the scientific rationale. At least for the second one, the blindness one. Could you expand on it a little bit? Is there enough rationale in the midst of uncertainty, scientific uncertainty, to think that this wouldn't be wasted money? Do you think that the power of genetics is understood enough? That's my first question for the blindness one. I can't remember the exact term here, the retinal organoid disease model. And the second is back to the HIV one. What is the goal of this project? Is it to cure HIV, or is it to perform better than antiretrovirals? In the first case, I think there's a scientific rationale. In the second case, I'm a little less clear. Thank you for your clarification, Gil.
Sure, absolutely. And so for the dominant optic atrophy, yes, there were concerns raised as it relates to the rationale of some reviewers stating, for example, I don't feel there's sufficient justification for using a retinal organoid model instead of further developing animal models. So I think the rationale was not related to the therapy itself, but generally the approach for using the organoid models versus using in vivo models. We don't have an opinion on the Serm team one way or the other. I mean, I think they were split in terms of that opinion. The majority of the grants working group felt it was fine to use the organoid models. Our assessment of this in terms of why we're recommending was really focused on whether, assuming that things work out, would they be able to achieve the goals of the DISC 2? And, there's obviously a risk for all projects, but we thought, yes, they could, assuming they would succeed in these studies, and that it would be difficult to address the most significant concern from reviewers, which was related to the underlying dominance mechanism, which we thought the applicants really would not be able to address in a resubmission. So those are the two things that really drove that one. For the HIV, so the goal is, for that one, is to develop a therapy that would prevent the latent reservoir from emerging. So as provirus emerges from that latent reservoir, there would be basically a mechanism of ongoing surveillance in order to prevent the virus from replicating. So it wouldn't necessarily be a cure, meaning it wouldn't necessarily eliminate the latent reservoir because it doesn't specifically target it, but the idea would be that it would prevent it from advancing or emerging.
Thank you.
Gil, can I add one thing?
Yes, please, Rosa, go ahead.
So with regards to the application with the one second, I was just looking. The first application, one of the comments from the reviewers was that even if we the project failed, the organoid model would bring a lot of value, because there is actually not, there's kind of a rift in the field as to whether organoids are a model that can work for testing in this disease, because the animal models are not good. So it could be reasonable spending at this level of funding, especially in DISC, to have as an outcome a really good organoid model that supersedes what the animal models can provide. So no matter what, we thought that this could bring value to the field. And that was answering Dr. Dilege's question as well.
Very
Thank you.
helpful. Thank you. Yeah.
Fred.
Again, just to be clear, the scores that we're looking at, the 15 people that voted for 13475 and the 14 people that voted for 13510, those were all the scientific reviewers. Those don't include the votes of any advocates or non-scientists. Is that right?
So the 7 versus 8 in terms of the score is only the scientific members. However, the grants working group votes against each criterion may include some of the patient advocate and nurse member votes.
Yeah. So again, I'm in no position to evaluate the science, but as a layperson in this process, I'm pleased to see that of the 7 people that scored 13510 at 85 or above, that the range went as high as 90. And having been through these meetings now, seeing that projects that have great enthusiasm score well above 85, and 85 is like the bottom in terms of confidence level, but people want to see it happen. And so when I'm looking at this, being asked for my opinion about it, seeing that the range included at least a 90 is helpful to see, and the Serm team's perspective about it is super helpful. For 13475, the range got no higher than 85, and the majority of the scientific viewers, a slim one, albeit, voted less than 85. So I'm having, just again, from a lay point of view, because I don't understand the science that we're talking about, from a lay point of view, I'm just cautious about going against the scientific reviewers, majority of whom voted that it should not be funded. And even those that voted to fund it voted with the lowest amount of enthusiasm for funding possible. So I just wanted to put that out there. Again, I'll rely on the recommendations of the Serm team about this. Obviously, they brought it to us and are recommending it for reasons. And I will, if this body agrees that these should be funded, I'll certainly vote in favor of that. But I just wanted to express what it's like as a layperson looking at this and making the decision based on the information that we have available.
Thank you, Fred. Any other comments from members of the board? I would just like to add a couple. One, with respect to the HIV project, the issue of the latent reservoirs has always been particularly vexing. And I think that something that aims to do something about that very serious concern is something that we should be behind, in addition to the other reasons articulated by Gil. On the dominant optic atrophy proposal, from a programmatic note, Gil, correct me if I'm wrong, but we don't have anything anywhere in the Serm portfolio on this particular indication. Is that correct?
Yes, that's correct.
So that to me, in addition to the reasons that Gil referenced as to why the team recommends funding that particular project, is something that leads me to support that notion. Any other comments? Do we have any comments from members of the public? Okay,
There are no hands. There are no hands raised.
Okay, thank you, Maria. So
JT, if I may, just for
Yes, Adam.
a point of clarity, the original motion did not state this specific applications that we are considering. It was just generically what the Serm staff recommended. So for clarity, can we just specify that the two are DIS 2 13510 and DIS 2 13475?
Correct.
Yes, thank you, Kevin.
JT, we do have public comment now.
Yeah.
I just want to confirm that this is public comment about the two applications that are currently being considered and not general public comment on other applications.
Okay, thank you.
Okay, so it says Jennifer Roslin, so we'll start with you. And it's limited to 3 minutes, so thank you.
Sorry, I think you were just confirming if the comment is related to the two
that are currently being voted on, correct?
No, my comment was not related to those two.
Okay, if you could wait until other public comment, we just would like to hear from members of the public on this specifically. Tiffany.
Yes.
Is this is your public comment related to these two applications?
So sorry. Stepping out of the operating room. I'm sorry, do what? Can you repeat the question?
Is your public comment related to either of these two applications? If not, there will be other public comment available.
Eva, can you assist here? No.
Okay, there will be opportunity for other public comment, and Anne Marie has her hand raised as well.
Anne Marie.
Thank you very much. Very useful conversation. Maria, Milan, I wanted to ask you a question in regards to the comment that Fred Fisher just made about the application on the retinal organoid disease model, the one that is a little bit more, on the cusp, really, and that would help me for my vote. Do you think that it is justified with the Serm that we support an application, not so much because we believe that it's likely to be successful, but because we believe that by doing this experiment, the entire field and that particular disease will understand better the mechanism of disease, in this case, the role of the genetics in the onset of the disease. Can you help us, guide our judgment here? Thank you.
So Anne-Marie, in response to the comment, which I think I understand Fred's question is, given the range of the scores and the pattern of the scores, does that speak to the strength of the application, and should that be considered, or was it considered, maybe I'm paraphrasing, when we made the recommendation to recommend this application under the minority report category? I will just start by saying that it's an imperfect science. We do our very best to give guidance and to reviewers, and Gil and team really do a great job with it. But when you're in that kind of the gray zone for scores, it's really not quantitative. So it really does rely on the aggregate information, both from the scientific review and putting that together. I think both Gil and Rosa, who, by the way, the entire science team comes together in this, really look at dissect the input of the GWG with regards to the application. In aggregate, we do believe that this should be funded based on the reviewers' comments and under the minority report umbrella. So, without going into that rationale again, for the rationale that was provided by Dr. Zambrano and reinforced by Dr. Aviles. Hopefully that's enough. And, whenever we have scores, I think what happens is it puts them in a category. So it allows for the discussion, and then it allows the board to make their programmatic discussion. Again, it's not an exact science, it's a guide to have those discussions, as the best way I can kind of represent, how scores are used. Anne Marie, I don't know if that completely answers your question.
No, but it does, Maria, and it helps me put together the comments from Rosa, Gil, you and others, that in that particular case, more than in others, while there's a lot of uncertainty about the value of the experiment to find a cure, it's always the case, particularly in stem cell research, there's even more so a value of this experiment to help scientists understand the field better. And for me, that counts as well. So I'll vote yes for this reason, to approve it.
Steve?
Yes, so let me make an additional comment regarding the one that involves the cell-derived retinal organoid model. So on June 9th, the House of Representatives, the US House of Representatives, voted to approve the FDA Modernization Act. The Senate has already approved that. I'm not exactly sure when it goes to Biden for his signature. But in that act is a requirement, well, I would say it's a requirement, but a strong push to have the FDA basically change the models that it requires using for testing for IND purposes, to move away from the animal model testing and to other mechanisms of testing. And one of those potential models is going to be the area of organoids. It's an up and coming area. So sort of a la what Anne Marie was talking about, but from a different point of view, if it doesn't actually work in this case, but help support the development of organoids as a model, I think just for that reason alone, it's worth funding, because we're headed in that direction. The use of animal models, and I know that was a critique in the assessment, but unfortunately or fortunately, animal models are at some point going to become a thing of the past, particularly when it goes to FDA for FDA regulatory purposes in terms of animal testing leading up to an IND.
Thank you, Steve. Any other comments from members of the board?
I just want to make one quick comment. We are at quorum right now, so if anyone needs to leave early, we run the risk of not being able to take votes. So I just want to make sure everyone's clear.
Okay, thank you. Seeing no more comments from members of the board, and Maria, I assume no more members of the public either with respect to these two particular grants, let's proceed now to a vote on whether or not to elevate these two grants as numbered by Kevin up to the recommended for funding range. Maria, will you please call the roll?
Sure. Leandra Clark Harvey.
Yes.
Anne Marie Dulege.
Yes.
Mark Fisher Colbury. I'm so
Yeah.
sorry. I was reading. Thank you. How about Fred Fisher?
No problem. Go ahead.
Yes. Yes.
Thank you. David Higgins.
Yes.
Steve Julesgaard.
Yes.
Rich Lahara.
Yes.
Lauren Miller Rogan.
Yes.
Adriana Padilla.
Yes.
Al Rolette. Marvin Souther.
Yes.
Jonathan Thomas.
Yes.
The motion carries.
Okay.
Just for clarification, Mark Fisher Colbury votes yes too.
Yes, I have that. Thank you so much.
Okay, thank you. So we now have those two added. We now have a total of 19 in the group recommended for funding. We do have two comments on other grants and the not recommended for funding range. I think this would be a time to hear those. So Maria, would you please invite those public commenters to speak to the board?
I think you just did. So if you have public comment, now is your opportunity. And again, it's limited to 3 minutes. And please just raise your hand and we can call on you. I know you're not Jennifer Roslin, but we're going to call you Jennifer for now.
Thank you,
Go
Maria.
ahead. You're
Yes.
welcome.
Thank you. Thank you, board, and thank you, everybody. So my name is Aijun Wang. I'm the PI for one of the 84s, number 3 on the list of with the minority report, 13413. I just have a very brief comment about this project. I actually, first of all, I want to express my great gratitude to the reviewers and also our Serm team on recognizing the importance and the innovation of the project. What we're trying to do, I really echo what the board member and Maria just mentioned, that we're trying to find a cure. So what we're trying to do in this project is really to trying to find a cure for a very bad disease, Duchenne muscular dystrophy. And what we actually have shown, very excited about for the preliminary data we have gathered, and many of the reviewers have also recognized, is that we found some special technology elements that can be utilized to target the heart and diaphragm in the fetus. Actually, before the baby is born, we can edit those cells in the diaphragm and heart. The reason this is important, because lots of the Duchenne muscular dystrophy patients would die very early, primarily because of the cardiac function dysfunction and also respiratory function loss. So this is a very, as one of the reviewers actually pointed out, it could be it's for a small population of a rare disease, but I think this goes along very well with our the Serm's motion. For example, recently Serm announced the partnership with the NIH and FDA and private sector to find cures for rare diseases. So I think this project, we are really excited about this, the new finding. We just don't want to get any delay on this project, but I really appreciate what Gil just mentioned, that even though we were ambitious, we're trying to complete the whole thing from the animal in vitro culture cell model to animal model to human disease model. It's a really ambitious team project, but we have a very wonderful team, and we also have an expertise in leading translational research into clinical trial. So actually, our team at UC Davis, led by Dr. Diana Farmer and myself, are actually conducting an ongoing clinical trial using in utero treatment for spina bifida. I know many of you may remember this project. Anyway, I want to say thank you to the team. If you could support this round for our project, we can move faster. But I would appreciate any comment or further consideration, even if we have to come back next round. It would just be a little bit delayed. But thank you very much.
Thank you. Tiffany?
Thank you so much. So our project is project number 13442, microgel encapsulated iPSC-derived notochordal cells to treat intervertebral disc degeneration and low back pain. So just to give sort of from the clinical side of things, some history, and why this is a huge project. So in March of 2018 in the Lancet, Dr. Harvickson stated that low back pain is the #1 cause of disability worldwide, with over 60.1 million years lived with disability in 2015 alone. Over a 25-year interval, the incidence of low back pain disability increased 54% worldwide, most likely due to our aging populations. Studies have found that in higher income countries, the incidence of back pain is around 33%, and the healthcare approaches for back pain contribute to the overall burden of disease and cost rather than reducing it. In lower income working countries, farmers decrease their workload to diminish the effects of back pain caused by manual labor, which also can increase that cycle of poverty in those areas. So the goal of our project is to figure out the ultimate issue with low back pain and degenerative disc. Our project wants to address the low back pain related to degenerative disc by exploring using pluripotent stem cells to revitalize and repopulate the nucleus pulposus. So what is the burden of low back pain in society? So the cost of medical care, indirect costs, productivity is lost, staying out of work, alternative medicine, and medications and time to and from appointments. The US alone has the highest cost for direct medical treatment and low back pain, primarily due to our medically intensive approach. Depression, chronic pain, opioid crisis, right? 500,000 deaths over a 20-year period attributable to prescription and illicit drug use of opioids. If we could find a stem cell treatment for low back pain caused by degenerative disc, the effects could be endless and seen worldwide. Psychological and mental health improvement, attendance in workplace, cost of healthcare driven down instead of up, productivity by farmers globally, and longevity of life accompanied by quality of life. Just this week at Cedars-Sinai Medical Center, where we are, the first human experimental injection of stem cells into the motor cortex for amyotrophic lateral sclerosis occurred. This is the first patient in the human trial for cranial injection of stem cells for ALS. Cedars-Sinai is committed to stem cell research. Our team wants to follow in leading this global quest for stem cell therapies in spine. Our project is fundamentally oriented at using minimally invasive stem cell biological solutions to recreate the nucleus pulposus and rejuvenate the intervertebral disc, preventing the cascade of events that we've talked about. Thank you for your time.
Thank you, Tiffany. Okay, so do we have, Maria, any more public comment on any applications that are currently not in the recommended for funding range?
Yes, we have a hand raised, and that is Lana.
Yes, hi, good morning. I wanted to clarify that the call for public comments was only for the grants that received the minority report, or is it any of the projects that were not recommended for funding?
Any of the projects.
Great. I would like to take an opportunity to make a comment then on project 13502. And it's just the one below the ones that received the minority report, titled Excitatory Spinal Interneurons from Human Pluripotent Stem Cells to Treat Spinal Cord Injury. Is it okay if I make the comment?
Yes.
Okay. Well, thank you for the opportunity to speak with you all today and briefly discuss my disc proposal to Serm. I would first like to express my gratitude for the reviewers of our proposals. I was very honored by a number of positive comments of their recognition that the proposal has potential for high scientific impact, was built on excellent preliminary data, and built on a solid rationale based on the work that we have previously published. And in line with the DISC-2 program, the research described in our proposal aims to identify a therapeutic candidate that could potentially help not only the thousands of Californians currently living with a spinal cord injury, but the hundreds of thousands of Americans living throughout the country, and of course, many that are overseas. And while the mean score of the proposal was 83, it's just below the funding. My purpose here today is really just to take the opportunity to address what looks to be the only one key score driving factor that dampened enthusiasm for our proposal. And that was a single concern from a couple of the reviewers for my personal professional experience to date. And I'm glad to have this opportunity to try and clarify this for the discussion. So there was one comment that I was not a contributing factor on papers that led to the proposal, which is not entirely accurate. And to clarify, the present proposal is built on prior work by myself and payroll work from my prior mentor, Dr. Todd McDevitt, of which this is a resubmission. And I was actually recruited into the McDevitt team at Gladstone Institutes and took over the spinal cord injury program to gather pilot data for the present proposal. And I've been leading this program for several years now and in charge of the dedicated funds and personnel. Second, there was some concern that I had little experience managing a research program of this size. And I really just wanted to note that in taking over the spinal cord injury program at Gladstone Institutes, I have helped bring in around $400,000 in funding to support and build the program. And it is now from this that I would like to build further with the present proposal of identifying a possible therapeutic target that we're very excited about and I feel prepared to manage. And I'm very pleased to have support from many senior scientists at Gladstone, including the president of our institute, Dr. Deepak Srivastava, who provided a letter of support for myself in this proposal. My prior mentor, who's now has left Gladstone, and he is at Sana X Therapeutics, leaving me in charge. my colleagues in the field of spinal cord injury and transplantation research, clinical professionals that volunteer their time to meet regularly, and individuals living with a spinal cord injury, such as Roman Reed, who's an ongoing supporter of our work and who also meets regularly with us. I really hope that I could clarify some points and now alleviate some of the minor concerns that some of the reviewers had. And I thank you for this opportunity to meet and speak with you, and thank you for the work that you are doing.
GT, you're on mute.
Sorry. Thank you. Deepak, you're next.
Thank you, John. My name is Deepak Srivastava, and as Lana mentioned, I do serve as president of the Gladstone Institutes, and I thought I would just make a comment on Lana's suggestions. The major feature that lowered the score out of the fundable range was, in fact, this concern about experience. And I should say that Lana seems to me to be an extraordinary talent who I've been impressed with how prominent her recognition is already in the spinal cord injury field, as I've come to understand it. And if there were, after Dr. McDevitt left Gladstone to go to SANA, Lana has really single-handedly led the effort, and we've been able to raise philanthropic dollars that she has directed to get to this point for this application. And this funding this application, I think, would really accelerate the work in spinal cord injury that had very positive scientific recommendations. In addition, if there's any concern about institutional commitment, then I can certainly address that from my role, in that we are committed to this area, to the stem cell area in general, as you all know, but Lana specifically and the spinal cord injury work that she's doing, I couldn't be more excited about it. And at Gladstone, we're committed to supporting it. Thank you so much.
Deepak. Maria, do you want to call the next guests, please?
Yes, I believe it is Jean Paz.
Hello. Can you hear me?
Yes, we can.
Yes.
So I would like to thank Serm and all the reviewers for a careful review of our proposal. The proposal number is DISC 2-13533, entitled Gene Therapy Vector Correcting Endoplasmic Reticulum Stress and GABA Uptake Defect in Myoclonic Atonic Epilepsy. Our DISC 2 proposal focuses on developing a desperately needed treatment for a rare, devastating neurodevelopmental disorder called SLC6A1 syndrome. This gene regulates brain development and excitability, and children with mutations in this gene suffer from autism, debilitating epileptic seizures, sleep, attention, and motor deficits. SLC6A1 mutations can cause death in children. My co-investigator Navneet Matharu and I are actually involved with the SLC6A1 patient organization, and we know the kids for whom we are developing these therapies. Developing and validating treatments for FDA approval requires an animal model. And our SLC6A1 Connect team recently developed the first mouse model that our lab was the first to characterize. This mouse model has severe seizures.
Next slide.
My co-investigator and I, Dr. Matharu, were approached by Serm in 2021 to submit a proposal on developing a gene therapy for this disorder. Sorry, can you hear me? I'm hearing some background discussion. Okay. So after discussing the project, we were advised to apply for the DISC 2 funding opportunity, defined as an opportunity for early stage projects that are high risk, high reward. And we submitted a proposal to get funds to develop a new vector during the first 6 months of the proposal, and then to use this vector in our mouse model that has a mutation that was discovered in a child with the disorder. In the proposal, we documented the feasibility of the approach and a careful characterization of the first mouse model of this disorder. Again, I want to emphasize that this disorder cannot be studied in organoids or cells. You do need a mouse model that has seizures in order to get an FDA approval later on. And we have that first mouse model. So during the first review, we got a score of 80. The reviewers expressed a significant enthusiasm, and they all emphasized that this could be really life-changing if this worked, and that it was a good proposal for a high-risk, high-reward program. However, the main concern was that we didn't have the vector yet, but we needed funding to get the vector. And after discussing with Serm in 2021, we were told that if we already had validated the vector, we would not be applying for DISC 2, we would be applying for the next step, which would be the preclinical translational step. So we were given—
You've exceeded the 3 minutes, so if you could wrap that up, that would be great. Thank you.
So we were given 3 weeks to revise, which was not sufficient to make the vector. We feel like the grant was reviewed as an R01 grant from the NIH rather than a high risk, high reward. And I think my collaborator, Navneet Matharu, is here, so maybe she can say a few words too. Thank you.
Next public comment is from Michael Lane. And again, as a reminder, you have 3 minutes.
Hi, thank you very much for giving me the chance to talk today. I'm just going to be making a comment on Dr. Jolodiva's proposal, which is just looking at the number real quick. I don't see it on the list anymore. But to follow up on the comments that both Dr. Jolodiva and Dr. Srivastava made, I wanted to firstly say I'm a collaborator on this proposal, and it gives me a great deal of pleasure to be a part of this work. The work that Dr. Jolodiva has proposed in this proposal is essentially building on iPS therapy, cell therapy for spinal cord injury, as she alluded to. But this is actually something that she has developed over the last several years, which has reshaped the way that cell therapies are being used to treat neural injury and disease. And there's a big shift in the field of spinal cord injury, ALS, and the field of transplantation for treating neural injuries and disease, which has essentially resulted from Lana's prior work, where she's now developing cellular engineering strategies to make cells specific for repairing injured substrates. And this is something that has not been done in the huge amount of work that's been done over the last several decades. I myself have been working in the field of spinal cord injury for more than 20 years now. And this recent shift in thinking as to how we can use cells to more effectively regenerate injured nervous tissue is essentially building off the work that Todd McDevitt has done and in the work that Lana Jolodiva did in parallel. I also want to commend the Gladstone Institutes for providing a great deal of support for up and coming rising superstars in the field of spinal cord injury, in the field of neural injury, like Dr. Jolodiva. And the support that Gladstone essentially has given her has positioned her to become a future leader in the field of spinal cord injury. And essentially in now running the spinal cord injury program at Gladstone, I look forward to being able to work with their team and assist them in any way I can. Thank you again.
JT, you're on mute.
Thank you. Yes, thank you very much. Maria, do we have an
Yes,
additional
Michi,
comment?
I think, is next, please. And as a reminder, you have 3 minutes.
Thank you. I'll turn my video, but so I just want to mention a few points that I integrated in my letter to the board, and I'm talking about application that Tiffany described, 13442 for the intervertebral disc degeneration and regeneration. So thank you, Gil, for the summary of the minority report. Just one point from that report, that the low scoring panelists were talking about lack of or insufficient amount of preliminary data. And what I just want to mention is that our previous Serm grant on this topic, we were able to show a feasibility of notochordal cells differentiation, and we showed them in a very translatable large animal model. And in this proposal, we actually trying to improve the formulation of the delivery system. And the goal of this study is to find a therapeutic candidate with these cells and in the final formulation. So when the reviewers are asking for final formulation, I think this directly speaks to the target of this program, the Quest program, to find the final candidate. Thank you.
Thank you, Dimitri. Are there any other public comments, Maria?
No, there are no hands raised.
Okay, I'm going to entertain a motion that we approve the 19 projects currently in the recommended for funding range. And if there's anybody who doesn't think that list is complete or whatever, this is the time to say something. I'll remind you that the two that were added were the only two that the team recommended we currently move up, and also note that the next submission date for the next round, which will be a 3-year round, is going to be August 2nd. So it is in the relatively near future. Can I have a motion that we approve the 19 projects currently in the recommended for funding range?
So moved. Seconded.
Okay, Maria, did you get that? There were about 5 so moves in there.
No, I did not. So if someone
Leandra.
could.
Leandra moved and
Fred seconded.
Fred seconded. Okay, thank you. Is there discussion by members of the board on this motion? Okay. Is there discussion by members of the public? And remember, this is about approving the 19 projects listed in the recommended for funding range. Maria?
There, let me go through the vote. I want to make sure I capture everything. Okay. And as a reminder,
Right. Yeah, Maria, before you say anything,
sorry.
You reminded me previously, which I was remiss in doing. Is there any motion to remove any of the projects recommended for funding from the recommended for funding range?
That's a different motion, JT. So do you want to entertain that motion first?
Well, I just wanted to see if there was a motion. It doesn't sound like there is one. So do we need to?
Oh.
Yes, I was
Kevin,
going.
from a procedural standpoint, I leave this up to you.
Yeah, from a procedural standpoint, we have a motion on the floor. If that motion were to be withdrawn, that's with the permission of the mover and the second.
Okay, well, I don't think that's necessary because there was no indication of any interest in the subsequent motion, if that's okay, Kevin.
That's fine.
Okay, so Maria, will you please call the roll on the motion to approve the 19 projects currently in the recommended for funding range?
Yes. And as a reminder, you can vote yes or no, except for those with which you have a conflict. Leandra Clark-Harvey.
Yes.
Anne-Marie Dulege.
Yes.
Isabel Dudon.
Yes, except for those with which I have a conflict.
Mark Fisher Colbury.
Yes.
Fred Fisher.
Yes.
David Higgins.
Yes.
Steve Julesgard.
Yes.
Rich Lahara. Rich, are you on the phone? Lauren Miller-Rogan.
Yes.
Adriana Padilla.
Yes.
Al Rolette.
Yes.
Marvin Southard.
Yes.
Jonathan Thomas.
Yes.
Art Torres.
Aye, except for those which I'm conflicted.
This is Rich. Are you guys able to hear me?
No, now we can.
Okay, sorry. So yes.
Thank you. The motion passes.
Thank you. Okay, the only remaining order of business on the votes is, do I have a motion to not approve those projects that remain in the not recommended for funding range?
I motion.
Moved by Anne Marie. Do I have a second?
Second.
Is there any discussion by members of the board? Any discussion by members of the public? And I would remind those who spoke, first of all, thank you very much for that. But this would not be a time to speak again on that, just to reiterate those points. Yes, Fred.
Apologies, could you just repeat the motion?
The motion is to not approve for funding those projects that remain in the not recommended for funding range, which are those in the white on the posted spreadsheet on the screen.
Thank you.
Any other comments or questions by members of the board? Any comment from members of the public? Maria, will you please call the roll?
Yes, and again, if you could say yes or no, except for those with which you have a conflict. Leandra Clark-Harvey.
Yes.
Anne-Marie Dulage.
Yes.
Isabel Dudoni.
Yes, except for those with which I have a conflict.
Mark Fisher Colbury.
Yes.
Fred Fisher.
Yes.
David Higgins.
Yes.
Steve Julesgard.
Yes.
Rich Lahara.
Yes.
Lauren Miller Rogan.
Yes.
Adriana Padilla.
Yes.
Al Rolette.
Yes.
Marvin Southard.
Yes.
Jonathan Thomas.
Yes.
Art Torres.
Aye, except for those of which I'm in conflict.
The motion carries.
Okay, thank you very much, Maria. Thank you, everybody, for that robust discussion and consideration. Thank you to all those who commented. Would encourage those of you that did not get awards today to reapply if that's the appropriate move on August 2nd. That concludes the Application Review Subcommittee portion of the meeting. Is there any public comment on items in general?
There is none.
Okay, hearing that concludes today's business. Thank you all. I want to remind everybody we have a meeting of the full board on next Monday and look forward to seeing you all then. Till then, have a great rest of your week and weekend. And Maria, thank you for all your work in navigating through this, as always, and to everybody on the team for their help.